All right, good morning, grade 12s. It's time to start our genetics unit. Always a lot of fun thinking about why you have the traits that you have, where they came from, and in honor, of course, I'm wearing my ATGC nucleotide geek shirt, and it is May the 4th, so May the 4th be with you. And let's get started. All right, so here we have a lovely picture of Gregor Mendel. We'll talk about him in a minute. He's the father of genetics, and he loved peas, just like I do. All right, okay, wow, let's figure out how we're gonna change the slides here. All right, great, here we go. So a few things we're gonna talk about. We're gonna talk about traits, of course, and you've heard that the terms dominant trait, recessive trait. We're gonna be studying some pedigree charts, incomplete dominance, multiple allele situations, dihybrid crosses, monohybrid crosses, probabilities of having traits that you have, talk about selective breeding, getting things that we want in gene interaction. All right, as promised, here we go. Here's Gregor Mendel, 1822 to 1884, so we're talking about a couple hundred years ago. He was an Austrian monk, and he made the first rigorous quantitative observations of inheritance patterns, proposed plausible explanations for his observations. I told you he loved the pea plant, Pisum sativum, because pea plants could reproduce sexually by cross or self-pollination. So it allowed Mendel to control the percentage of successive generations. All right, and he determined, and we'll talk about his laws in a minute, that he determined for every trait there must be two governing characteristics. He called those factors. We'll call them alleles, one coming from the sperm, one coming from the egg, and we can also call them genes. We'll get into the vocabulary quite a bit, guys, today. You're gonna learn some geno-speak, all right? Let's raise this light up I'll bet, so I don't look too ghostly and what have you. All right, and he discovered some specific laws and principles of genetics. So here we go right here. We have this thing called the principle of unit characteristic. That means for every characteristic, you get two alleles because you have two parents, all right, at least biologically. He also determined, and like I said, we're gonna call them alleles. He is the one that we can give credit to the principle of dominance, that one trait has a dominant form that might be expressed over another form, and if the two alleles differ, one only would be fully expressed and the other is masked, the recessive one, of course. He also determined the law of segregation. So when we did our study on meiosis just recently, we know that during gamete formation, the pair of genes that are responsible for each trait, the maternal one, the paternal one, they separate or segregate during the formation of those gametes. And we also have this thing called the law of independent assortment, which kind of says that for each allele pair, they're going to separate independently. In other words, for Mendel at least, and for the pea plants he was studying, he kind of lucked out that his alleles or genes were not tied to each other. So there you go. All right. Now, some vocabulary as promised. So we're talking about traits. Traits are characteristics of an organism, such as the color of a flower or the height of a human. We're going to have this term phenotype. Phenotype in genetics is how the trait is observed. I often think phenotype like physical. It's what we actually observe or see. Yes, okay. Now, what is a gene exactly? So we can't see the genes of an individual, but the gene is what we call the functional unit of heredity. It's an ordered sequence of nucleotides. So remember those nucleotides that you have studied in science, maybe even as far back as grade 9, but we have these 4 nucleotides, A, T, G, and C, and that forms the genetic code of your DNA. The combination and number of these nucleotides form what we call the genetic code, and a segment of that code that codes for something like a protein is considered to be a gene. All right? Now, we've already mentioned the term alleles. Alleles are the forms that a gene can take, and each allele is found at the same position on each of the homologous chromosomes. Remember that we usually have 2 alleles for each gene, one that came from mom and one that came from dad. I often like to think of alleles like like flavors, right? So if it was ice cream, for example, that you were after, the different flavors are kind of like the alleles of a gene, right? So you can have a gene for tongue rolling, common quintessential trait that we often talk about, whether you can roll your tongue. I can roll my tongue, all right? By the way, that's a phenotype because that's the physical observation you make, right? So tongue rolling is the trait, and I can roll my tongue, so I'm a roller. That's my phenotype. But what alleles, right? What forms of that gene do I have? I have two forms, right? There's two forms. There's the rolling gene and the non-rolling gene. The rolling gene is dominant. The non-rolling gene is recessive, all right? So just like ice cream, like ice cream as in the thing, a trait, there's different forms or flavors that it can take, all right? Chocolate, vanilla, right? Rolling, non-rolling. Of course, chocolate would be dominant, all right? So as I said, the dominant allele is the form of the gene that you would see, right? So in tongue rolling is dominant, and that's the one you're going to see if it was paired up with the non-tongue rolling gene. The non-tongue rolling gene gets masked. Now, in genetics problems, we assign alleles a letter quite often. Doesn't always have to be a letter, but quite often it is, especially if it's what we call Mendelian genetics, all right? And we would give the dominant allele a capital letter, and we just arbitrarily kind of decide which letter of the alphabet we're going to use. So if we're going to do tongue rolling, for example, we might pick R for rolling, right? And because rolling is the dominant form, so capital R would be the allele for the tongue rolling gene. The recessive allele is the allele that gets masked by the dominant form. We're going to keep the letter, right? So we're not going to change the letter. We're going to keep R for the tongue rolling trait. Capital R means is the rolling allele, and a little r. So we use a lowercase letter for the recessive. So that would be not being able to roll your tongue, the gene for that. All right. Now, we talked about phenotype being the physical expression, the physical observation. I can roll my tongue. But what my genes are, what alleles I carry for that trait, that's expressed as what we call the genotype. So it's the combination of alleles that someone happens to have, okay? Pure breeding. So guys, the term pure breeding is used especially with plants. That means that when the plants are self-pollinated and they produce offspring, they're going to have traits that are identical to the parents. So that's where we get our term purebred. I mean, we use that with dogs too, right? And so if you're gonna buy a purebred golden retriever, you're expecting that both parents were golden retrievers and had those traits. We give the parental generation, when we're doing genetic problems, we give the parental generation a capital P, all right? And that would be our parents. The offspring of the parents, we call the first filial generation, and we give them the symbol F1, all right? So sorry, we're not talking about Indy car racing here. We're talking genetics, all right? So F1 would be the offspring of the P generation, and if it's plants we're using, we could actually do what we call a self-cross or self-fertilization. Now, if we have another generation after that, we would call those the F2 generation. So these would be the parents of the F2, grandparents, right? All right. Now, homozygous. You see the root here is zygous, so this would refer to, hey, what alleles do we have in the zygote, right? And if it's homozygous, that means that the alleles are identical for whatever trait it is that we're looking at. So I know the slides are using bees, but if I go back to my tongue rolling example, a homozygous situation would be having two capital R, capital R. Remember, we always have two, one from mom, one from dad. So remember, I can roll my tongue, so I could be homozygous. Dominant, capital R, capital R. Now, another way to be homozygous would be homozygous recessive. little r, and if you only have non-tongue rolling alleles, you would not be able to roll your tongue. All right? Now, another term we have is heterozygous. So this is when you have one of each form of the gene. So two different alleles. In the slide, it's big B, little b, but for tongue rolling, it would be capital R, little r. Can that person, are they a tongue roller or a non-tongue roller? Well, remember, they have one dominant allele. That's all it takes to have that trait. So I could be capital R, little r in my DNA, or I could be capital R, capital R. How would I find out maybe what I am? Well, we'll kind of talk more about that later. It's when we get into genetic problems. All right? Okay. Now, why are some genes dominant and some recessive? So if we consider albinism, for example, a condition where the individual cannot make the pigment melanin, which gives us our skin tone, we can see what is happening. If an individual has at least one copy of the gene that enables them to make all the enzymes to produce melanin, they will be normally pigmented. So you just need one gene, because it's dominant, to make melanin. So in this case, let's call that capital A. So that's the allele that allows you to make melanin and have skin tone, right? So you could be, remember the terminology here, homozygous dominant, both capital A's, or you could be heterozygous, have one of each. Either case, you have what's necessary to make melanin and have skin tone. But the albino individual has a mutation. So guys, mutations are often what causes there to be recessive alleles. But you'd have to have a mutation in both of your copies of that gene so that neither one has the code to make the enzymes that are necessary to make melanin. Their genotype, of course, will be homozygous recessive, little a, and then they don't have any melanin, so they are albino. Okay, so we see even in goldfish has pigment because they have at least one dominant gene to make pigment. An albino goldfish doesn't have the allele to make pigment, so they're homozygous recessive. Now, here's some terminology. If we have a person that has the normal, typical allele to make pigment, but is heterozygous, we often call them a carrier, especially if it's a mutated allele. They carry the mutated allele, but what are they going to look like? This fish right here, because it only takes one dominant allele to make the color that's necessary. All right, let's make sure you can see here okay. All right, now Mendel used pea plants. And why did he love peas so much? They're small, so you can get a large number of peas very quickly. They are kept in a small space. They have a short life cycle, so you can get a lot of generations very quickly. They can self-fertilize or cross-fertilize because they're typical plants. Plants can do that because they're hermaphroditic. They have both male and female sexually reproducing structures. And peas exhibit a number of visible inherited traits. So traits that are easy to see one way or the other, and they can only be expressed in two different ways, like tongue rolling, right? I can roll my tongue or I can't roll my tongue. There's no halfway, all right? And peas have a lot of those traits. Tall, short, yellow or green seeds, purple or white flowers, etc. There's a whole list of them there, all right? All right. Okay, now guys, remember, these are all phenotype terms, right? I see green pea pods, right? We have flower, so those are all considered to be phenotypes. Now the genotypes start to fill in here. We could assign, often we assign, get out of here. Often we assign, maybe the dominant trait often gets the letter designation for that trait. So yellow seed color is dominant, so hey, we could give a capital Y for that trait, or Y is the letter for that trait. So if we have a capital letter, we know that's the dominant allele, that's homozygous dominant. That's the heterozygous condition. Either way, you get yellow seeds. The only way you get green seeds, of course, the only way you see the recessive trait is homozygous recessive, both little alleles. All right. Oh no, now what did I do? That's bad news. Maybe if I go to the next slide, it'll sort itself out. No. Okay, there we go. Back to business. Right, now guys, we're gonna talk about something very important. A geneticist's friend is a Punnett square. So this is a tool that allows us to predict genotypes or phenotype or phenotypic ratio of offspring if we know the genotypes of the parental generation. So to do this, we use a Punnett square. I have awesome videos, okay, by the Amoeba Sisters. They do a really good job. So make sure you check out Amoeba Sisters videos on monohybrid Punnett squares, that's looking at one trait, dihybrid Punnett squares, when we get to those, 2 traits, etc. All right, so how do Punnett squares work? They are charts, right? And they help us predict outcomes. What we want to know is phenotypic ratio. So that's the ratio of the offspring with the dominant trait compared to those that have the recessive trait. Remember, what we can see. And that's usually what we want, the most important thing we want to get from Punnett squares. We can also get the genotypic ratio. Remember, that's looking at the alleles themselves. And we could kind of see the ratio of homozygous dominant individuals, heterozygous, homozygous recessive, right? Here's somebody that loves genetics so much they have a tattoo, right? So remember that, see all the alleles. So let's get into how to set up a Punnett square. So guys, when we set up a Punnett square, we're gonna do monohybrids first. So monohybrids looking at one trait. So if we're talking about Mendel, maybe he first crossed pure breeding round pea plants. So they produced round, nice smooth round peas. Well, if they're pure breeding, they only carry the capital R for round seeds, round peas. And he crossed them with pure breeding wrinkled peas. And so pure breeding wrinkled peas would only carry the recessive allele, the little r. Now when we set up a Punnett square, we make a grid pattern here. And essentially what we do, remember P is for parental generation. There's my one parent plant that's purebred for round. The other parent plant, purebred for wrinkled. We put this X, meaning we're gonna cross them together. We're gonna cross these parents. Now what we do, oh, I did that again. I don't even know how I do that. All right, there we go. So, guys, when we set up a Punnett square, we separate the alleles because remember, we are, what's the word? We are making gametes, all right? So remember, if we're gonna have offspring, we've got to create gametes. So we're sort of mimicking what gametogenesis does or meiosis does. It separates, remember we have to separate. So we take one parent and we separate the alleles and we put them there. We take the other parent, separate the alleles, and then so this could be maybe the sperm, right? Male. Maybe this is the female. In humans, that would be the eggs. And so this would mean that this sperm finds this egg and we simply combine the alleles in the boxes. We tend to put the capital first, that's just convention, all right? And so that becomes the genotype of a possible offspring. That would be heterozygous. Now you just happen to notice that that is true for every offspring. There's only going to be one genotype possibility for that offspring. Capital R, little r, heterozygous. What is the phenotypic ratio, or what's the phenotype? What do we physically observe? Well, this offspring, this plant, would be producing only round seeds. Only round seeds. We'd have only round offspring. Only round peas would be produced by that cross. Okay? Now, guys, there would be nothing wrong, and I don't know what's going to happen here. There would be actually nothing wrong. This is a bit, yeah, I thought that might happen. Wouldn't be able to write on my whiteboard. But guys, there's actually nothing wrong with doing what we would call a 1-by-1 Punnett square. If you can obviously see there's only one possible allele for the gametes, for one parent or both parents, there's actually nothing wrong with doing a shortcut and just having each different allele. And then you'd only have one possibility, and that's okay to do that. All right. There we go. We talked about the genotypic ratio. We talked about the phenotypic ratio. Only one possibility. Now, what if we looked at the next generation? So if we look at the next generation, we're going to take our F1 plants and we're going to cross-fertilize them. So remember, each plant we just saw was big R, little r. So we're going to cross those plants together. All right, so remember what we do? We take one parent and separate the alleles. We take the other parent, separate the alleles. We fill in the boxes as if each gamete were to combine. Now we get some variety in this offspring. In the F2 offspring, the second generation, we're going to get some variety. We can see that 3/4 of the offspring are round. Remember, that's a phenotypic ratio. 3/4 of the offspring are round, and only 1/4 of the offspring wrinkled. See, so that's good information, all right? We have done some predicting, and sure enough, remember, these are predictions. If I actually got a whole bunch of peas and I started to separate them, this is what I would expect. In reality, it might not be exactly that ratio, but it's what we expect. So the genotypic ratio, not that we care too much about the genotypic ratio, would be one round, one homozygous dominant, 2 heterozygous genotypes, and 1 homozygous recessive. But it's the phenotypic ratio that we are usually more interested in. We should get a 3-to-1 ratio of round to wrinkled seeds. Now, Mendel found that the recessive trait didn't disappear entirely. It reappeared in a later generation. All right, and that law doesn't only apply just to peas, but all to other diploid organisms. So guys, often recessive traits skip a generation, right? In the parents, we only have round, and then in the F1, it seemed to vanish. We had no wrinkled, but then when we did the F2, we ended up seeing wrinkled seeds. All right, so this is just the exactly what we were talking about, done with actual peas. There's our pure breeding parents. So you can spend some time on this slide in the slideshow if you like, but in order to keep things rolling, I'm going to keep rolling. Okay, speaking of peas. All right, we'll come back to the assignment in a minute. Now guys, I'm going to cover one more thing before I'm done this morning. Now, since homozygous dominant and heterozygous individuals look the same, how do we know for sure which is which? So for example, you knew that I could roll my tongue, correct? That's the dominant trait. That's my phenotype. But do you know if I'm homozygous dominant, capital R, or am I heterozygous, capital R, little r? You don't know my genotype, so how do we know? Remember, I kind of talked about this before. We know because we could do a test cross. So test crosses are crosses with a homozygous recessive individual. So guys, that's really important to remember. If you're going to do a test cross to figure out what the genotype is of something with the dominant trait, you do a test cross always with a homozygous recessive individual. So in my situation, not that we do this a lot with humans, but let's just say, if we knew that my wife was a non-tongue roller, she'd be perfect to do a test cross with because she would be homozygous recessive. Then we could look at our kids. Now we'd want to have a lot of kids. More kids we have, the more offspring we have, the better we can identify what my unknown genotype is. All right? So fortunately, we're not going to use humans. We're going to talk sheep here for a second. Now in sheep, black wool is more brittle and difficult to dye, so white wool is more desirable. And white is the dominant trait. So we could assign capital W for that dominant allele. Black, which we don't really want, which is a recessive mutation, is a little w. All right? Now let's say we have a white ram, but we want to know, because we're going to breed sheep, and we want a pure breeding white parent, right? So we don't know if that ram is homozygous dominant, big W, or heterozygous, big W, little w, right? Now, so what we do is we do a cross with a black ewe female sheep, because we know that female sheep will be little w. So what we do is, we know for sure, remember, we could look at two different Punnett square situations. Little w, because we know that's the black ewe. And these are the possibilities for the ram. The ram, if he's homozygous dominant, is cap WW, caps. If he's heterozygous, he's got one of each allele. This is the expected ratios. Notice, if the ram is homozygous dominant, there will only be white sheep, even though the female is only carrying black alleles, because this dominant allele for white dominates, because that's what it does. Every lamb will be white. However, if our ram is heterozygous, we notice that 50% of the offspring are expected to have the black phenotype. So as long as we have enough lambs to look at, matter of fact, we only have to see one, there he is, black lamb, we know where that allele came from. That one allele had to come from dad, and dad is not pure breeding. So you might not want to, use him for breeding if all you want is white wool, right? And you don't want to buy him necessarily because he's not purebred. Okay, so, and again, this cross I think is in your textbook, right? Same kind of idea. Excellent. Okay, that's as far as we wanted to get today. So guys, make sure you do your genetics problems. Got to do the problems to make sure you understand and know what you're doing. All right. Thank you for listening.